The overall goal of this electrophysiological protocol is to provide the ability to measure the kinematics of eye movements of the red-eared slider turtle using an in vitro isolated head preparation.
This method can help answer key questions in the oculomotor field, such as how vision evolved in turtles. The main advantage of this technique is that it does not require training animals to track targets and uses a gimbal to calibrate their eye movements. Demonstrating this technique is Stephen Nesbit, an undergraduate student in my laboratory.
Begin with the turtle head in a dissection dish. Have enough turtle ringer solution on hand to irrigate the tissue. Maintain the tissue at 4 degrees Celsius by placing ice around the outside of the dish. Place a blunt dissection probe through the mouth to provide easier handling of the head. Then, while working under a dissection microscope, cut the joint connecting the dentary bone to the cranium with the scalpel. Next, use rongeurs to pull the lower jaw away from the cranium. Then pull off the skin and muscles from their attachments at the dorsal and lateral regions of the cranium. After identifying the vertebral column at the caudal end of the cranium, bend the vertebral column ventrally to expose the spinal cord, and use microscissors to snip the spinal cord. Use rongeurs to remove the vertebral column and other tissue from the cranium by pulling caudally.